All right. Hi, everyone. Welcome to the last and best QLS seminar talk for this term, for this academic year, rather. Our speaker for today is Jonas Lehnert. He's from University of Ottawa. Jonas actually was a McGill student prior to joining Ottawa. He was doing his PhD with three parents, Eric Cook, Arjun Krishnaswamy, and myself. And he's been really amazing. He joined us in 2019, and he graduated just last fall, December 2025. Yeah. So I'm looking forward to hearing your talk.
All right. Just make sure. So you put expectations pretty high for the last seminar. Anyway, thanks all for being here. It's really great to be able to present in the same seminar series that until not so long ago I was still a part of. So thanks for CAMBAM and QLS for hosting me. The work I'll be sharing with you today is really founded in one of my major interests, which is states of mind. Every day we're surrounded by all types of sights, sound, and other stimuli. But based on our state of mind at the time, we may elicit a very different behavioral response. Our mood or goal, or even if we are hungry or didn't have a morning coffee, seem to have a powerful influence on how we respond to the same type of sensory stimulus on a daily basis. Now, it's widely believed that this process of external information, as it's married with internal state information, like our mood, gives rise to perception. And today I'll talk about one such perceptual process, where we place more importance on some visual stimuli than others, based on behavioral relevance, based on context, based on our internal states. This perceptual process is called attention, and it's been studied for many years now. And because of that, there's a couple of things that are really well established. Attention can be viewed as a spotlight that is placed at the most behaviorally relevant location in our visual scene to enhance our detection of the stimuli within it. Now, what decades of studies in non-human primates tell us is that these types of behavioral changes occur because the visual neurons that carry the information about the attended locations experience an enhanced gain in their responsiveness. Now, whether these kind of neural correlates are causal or how they are recruited is still not fully understood. So what is the attention signal? Where does it come from? What circuits govern it? And how do our internal states recruit it? Those were the types of questions that I wanted to answer when I started my PhD in QLS 7 or so years ago. And today I'll share with you what I learned. Now, before I begin, I first have to point out how hard it actually is to measure attention. So some of you may have experienced this when you're giving a talk. Or is the audience really paying attention to you? The indicators aren't really clear. And the reason for that is that attention is often covert. It's highly flexible, and it can be recruited at any given point in time as a voluntary process at any given strength. So with that in mind, in the first part of my talk, I'll share with you how we measured attention in mice, and then how we use that method to show that serotonin, an important neuromodulator in the brain that is thought to carry internal state information, seems to control how much attention the mouse brain, or the brain maybe in general, can deploy on a second-by-second basis. Okay, let's begin. It all kind of started with teaching mice to play this game. And in this game, we present mice with two screens rather, one on the right and one on the left. And we present randomly updating checkerboard noise on both screens, updating randomly at 30 Hertz. And the mice are tasked to detect the 3 right vertical bar grading that appears from that checkerboard noise at a random moment in time. Mice have to detect this grading to get their reward. Now, we can present this grading at different stimulus strength, which we call stimulus coherence. At high stimulus coherence, the 3 bars are easy to see. At low stimulus coherence, they become harder to see. The way that a mouse goes through an individual trial is that we first give it an audiovisual cue, telling it on what side of the screen will the 3 bars eventually appear. The mouse then starts a trial by licking on a lick spout in front of it, and then has to wait a random amount of time while searching through this checkerboard noise until the 3 bars appear and then respond to it again to receive its reward. And we call those hit licks. Now, in short, mice can perform this task really, really well. At high coherences, mice perform at about 100% of trials correct. At lower coherences, that performance drops as the stimulus becomes harder to see, like the one right here. And this was something that was true for many of the mice that we've trained on this task. Now, these types of psychometric curves have more interesting features about them. But first, I have to tell you about another feature in the behavioral task itself, which is what happens when mice don't wait long enough for the 3-bar grading to appear, and instead lick just in response to the dynamic checkerboard noise that they're supposed to reject. We call these false alarm licks, and when they happen, we simply restart the trial. We give them the cue again. The mice have to wait again for this until they fulfill this random amount of waiting time for the 3-bar grading to appear. And they can go through many restarts per trial, often restarting 4 or 5 times. Now, even the best trained mice performed a bunch of these false alarm licks. And so when we were designing this task, there were a couple of opinions in the room because nowadays a lot of people train mice to do behavior. One of the opinions was that, well, they're not going to wait long enough for the 3-bar grading to appear, which was like 10 to 12 seconds, because mice are impatient. But we thought otherwise because we designed this as an attention task. What turns out is that many of these false alarm licks occur because mice see something in the noise that resembles the 3 bars that they're searching for in the first place. The way we can show this is by first computing features from all of these stimuli that mice are presented with. For example, take a stimulus where there's no noise, just the 3 vertical bars. If you convolve it with a vertical Gabor patch, you get something called vertical energy. As you might expect, vertical energy or verticalness in this type of stimulus is really high. If you play the same trick with horizontalness, you get very little horizontal energy. Of course, you can perform this type of computation now on noisy checkers, where coherence, let's say, is 0.3. You get some vertical energy, or when coherence is very high, you can see the 3 bars in here. Vertical energy is also really high, with no changes across horizontal energy. Now, of course, you can not only do this computation when gradings appear, but also on all the single noise checker frames that precede these false alarm licks that mice so often form. And when you look at that, you find something surprising, which is that some 300 milliseconds just before a mouse false alarm licks, there tends to be this agglomeration of verticalness, vertical energy, whereas no changes happen so much in the horizontal energy. So could it be this that is actually causing mice to false alarm lick? Now, you can kind of test this by regressing changes in vertical energy towards the probability of a false alarm lick occurring by using a logistic regression. A logistic regression has a parameter beta, which basically governs the slope of this function, indicating how sensitive are mice in their false alarm lick decisions to changes in vertical energy. So in this case, that sensitivity is pretty high. And we refer to these betas as behavioral sensitivity. Now, of course, you can perform this computation not only in one spot of these checkerboards, but for every single checker in the stimulus. And what turns out is that you come up with a nice behavioral hotspot where mice are most sensitive to changes in vertical energy, whereas on the outside of these stimuli, mice aren't very sensitive to these changes. What's more is that if you play the same trick for horizontal energy, you don't get this relationship. These don't seem to elicit false alarm licks. And if you compare the cued screen versus the uncued screen, the relationship is much higher on the cued side. Mice are allocating their sensitivity to cued checkerboards, and as a result, you get more likely false alarm licks happening. Now, this was also true across many of the mice that we trained. You have this higher behavioral sensitivity in this 30-degree patch on the cued side of the screen that is much lower on the uncued side. No sensitivity towards horizontal energies. And this timing also matters because when you shuffle the order of these checkerboards, this relationship falls flat. It seems that mice are tending towards to the cute screen to detect these vertical changes in energy. Now, of course, you can perform this computation not only for verticalness and horizontalness, but all different types of orientations at all different spatial frequencies of these Gabor filters. You can also look at local contrast. How much white is there at that part of the screen where mice tend to be sensitive? It's dark. And what that allows you to do is to get a nice map of what features are mice searching for. At 0-degree orientation, mouse behavioral sensitivity tends to be very high, and at a spatial frequency that happens to be the same one as of the 3 vertical bars that the mice are searching for. Some mice only use these types of vertical features, while other mice also use contrast at this behavioral receptive field. And so this also happens to be true for many of the mice that we've trained. Here's an average of 13 mice, where sensitivity is always higher on the cued checkerboards and lower on the uncued checkerboards. So mice are tending, and this gives us a nice measure of how much they are tending. It's the difference between the central location here on our feature map versus on the uncued side. Now, please remember that this is not something that we explicitly told mice to do. These are false alarm responses simply by showing them noise. And what the mice tend to extract through their false alarm licks, which are not rewarded, are features that resemble the 3-bar grading that they're searching for. In fact, this is a strategy that persists all the way until they finally wait long enough through this delay period that the 3 bars exist. We can look at licks close to this time regime, and you find a similar relationship. And then lastly, it turns out that people have done these kind of similar tasks in humans, and the maps actually look quite similar. Even though the human visual system is so much better than that of a mouse, it seems that the attentional difference that you get by cueing and searching for rewarding stimuli is very comparable to that of mice. So maybe mice are a good model to study attention with. Now, there's many more things that we did to test this kind of behavior, but I just want to bring up one more of them, which is that this is a developed behavior. It takes time for this to develop. Mice train for many, many days on this task. Members of the lab know this too well. It takes about a month and a half for these type of nice sensitivity maps to actually develop, whereas in early sessions, they're not so strong. And what that allows you to do, this type of development of the maps, that you can take horizontal slabs or vertical slabs and plot, let's say, orientation tuning. How much more do mice become tuned towards vertical features, or how much more do they become tuned to certain special frequencies? Like this one right here, which happens to be the same one of the vertical stimulus that the mice are searching for. So to quickly recap, what I've shown you so far is attention task in which mice are tasked to detect these 3 vertical white bars that emerge from randomly updating checkerboard noise. They solve this task by allocating spatial sensitivity or attention towards cued features and locations, such as this hotspot right here, and such as features in this band, in order to make the most behaviorally relevant and rewarding decisions. How does attention do that? There's many models of attention, and I'm going to share a very simple one with you, where the visual circuits take in stimuli such as these, convolve them into features like a whole bank of Gabor filtered stimuli, and then sends them to decision areas in the brain to make rewarding decisions. It's attention's job then to reach in and select the most behaviorally relevant features, such as these spots right here and this feature set right here, and amplify them such that mice can make more rewarding decisions. Now, how this happens in the brain is still not fully understood. In mouse, we know very little, but we can take some inspiration from the decades of work that's been done in primates, which suggests that frontal cortical circuits, which form reciprocal connections with the visual areas of the brain, are one of the major culprits contributing towards attention behavior. Another culprit that seems more understudied are important subcortical nuclei, like the dorsal raphe, the locus coeruleus, and the basal forebrain, which emit important neuromodulators that signal internal state information throughout the entirety of the brain. Now, while most of the attention research has been focused on these frontal areas, little effort has been paid to these subcortical nuclei that emit important signals like value and reward throughout the brain. So could it be that they are involved in attention? And so what I'll show you next is some work that we did on serotonin, which is one of those important neuromodulators that seems to signal important internal state signals. Now, we focused on this molecule for three major reasons. The first is that serotonin lives in the dorsal raphe, a single accessible region in the brain that projects throughout the entire forebrain. Second is that when serotonin is applied to visual cortical neurons, you see effects that resemble those gain changes that I kind of showed you in the monkey cartoon earlier, where visual cortical neurons tend to respond differently. And then third is that many of the mental illnesses that are treated with serotonergic drugs often carry as a side symptom attentional dysfunction. So could it be that serotonin seems to somehow regulate attention in the brain? Well, to learn more, we formed the experiment again. We trained up a bunch of mice on the task. They learned really well. We had nice psychometric curves with very high hit rates at high coherences. The reaction time drops as you look for very easy to detect stimuli. And then we can use the false alarm licks produced by these mice again to compute our metric of attention. We again take these noisy checkerboards that precede false alarm licks and now compute the sensitivities. Now, one thing that our brain tends to do when we compare these maps on the cued and uncued side is a very simple thing. It's how hot is this blob on the cued side versus how hot is this blob on the uncued side. Second is, are they both using the same type of location? Are they both using verticalness, for example? Like in this case, they're always at the same location. So we tried to simplify our model a little bit to capture these two things more directly. And so in this updated model, we now map for different features these types of sensitivity maps, or in a downsampled way to make computation a bit easier. So you have a vertical sensitivity map and a contrast sensitivity map. Those are two features that are in the checkerboard noise stimuli. And then these are placed on both the cued features and uncued features, and are multiplied by two gain terms, either the cued attention gain or the uncued attention gain, all in effort to try to predict false alarm licks. What the modeling gives you is two things. The first is kind of gives you attentional focus. So on what part of the screen and what features are we using to predict false alarm licks? And the second, this weighting through the attention gains and the uncued attention gains. Similar kind of finding emerges where cued attention is always higher than uncued sensitivity maps. And if you take the difference between these, you end up with sensitivity maps weighted by attention. The kind of checkers tell you what features are we using. The redness tells you how much are we using these features on the cued side. And then secondly, what this gives you is a nice attention index. How much stronger are we using cued features versus uncued features by just subtracting these gains, normalizing them. And what you get is this attention index. So for all our mice, this attention index was positive, meaning that they preferred cued features and locations. Now, while we're performing these metrics of attention, we can also record from serotonergic neurons living in the dorsal raphe of these mice. We do this by expressing GCAM6F, which is a genetically encoded calcium indicator in the serotonergic neurons of the brain. And then we implant a fiber on top of the dorsal raphe to collect the light emitted from these neurons as they're activated. We do all of this simultaneously while the mouse goes through the different events of the task. And you get the serotonergic signal as mice go through start licks, the multiple false alarm licks that might happen in the trial, all the way until they either hit to detect and get the reward, or if they miss. Now, many of these types of events seemed important for serotonergic signaling, like start licks gave you a positive response, false alarm licks gave you a positive response. If they detected it and got a reward, they got a positive response. But what really caught our eye is the difference between hits and misses, trials in which the mice correctly detected the grading or where they seemed to miss it. During hit trials, just a couple of seconds preceding the grading onset, there seemed to be this dip in serotonergic activity that just wasn't also much there in missed trials. And I've sorted them by waiting time here. So in this waiting time, mice are searching through checkerboard noise. It's the same time at which they're deploying attention to solve this task. So could it be that these types of dips that happen in serotonergic activity are linked to attention? Well, to kind of test this, we performed a very simple analysis in which we first took trials where serotonin activity was low before grading onset or high before stimulus onset, or 3-bar grading onset. And what you end up with is these two trial chunks, both of which can contain hits and misses. So now you can compute psychometric curves again, and what you find is that mice performed better when raphe activity was low just before grading onset, but only at hard to see stimuli when the grading coherence was really low. There was no change at the maximum grading coherence when the trials are really easy, or minimum grading coherence when the grading was actually visible. Now, you can quantify these types of behavioral changes both by a commonly used metric, the discriminability index, how well are mice able to discriminate the 3-bar grading from noise. This was higher when raphe activity was low before grading onset, or the threshold, the 50% curve, signifying the shift in the psychometric curve here. Threshold was lower when raphe activity was low before grading onset, showing that mice are more sensitive to changes in grading coherence when raphe activity was low. What didn't change is again the maximum of these psychometric functions. Now, the maximum can be interpreted as like a metric of motivation. You can imagine when the trial is really easy and I'm not going to respond to it, then maybe it's because I'm not really motivated. These types of measures didn't change. What also didn't change is the response bias, where the grading was actually not there and I'm just kind of guessing to see if I can get a reward. Those types of behavioral strategies also didn't change across whether raphe activity was low or high before grading onset. What did change, right, is only the performance at these hard-to-see stimuli. This is exactly what attention can help you with, to enhance your performance at the behaviorally relevant stimuli. So could it be that that is what serotonin's kind of controlling here? Well, to do this, we can take false alarm licks from the same data set, partition them into was 5-HT activity low before a false alarm onset or high before a false alarm onset. And again, compute these sensitivity maps and then weight them by attention. What didn't change is what features mice use. They still use verticalness and contrast, whether or not raphe activity was low or high before false alarm onset. But what did change is how much they used them on the cued side. When raphe activity was low, attention towards cued feature in the patients was much higher than when raphe activity was high. It's also characterized by an attention index, which in all mice was higher when raphe activity was low before a false alarm. So it seems that we have this neural correlate of raphe or serotonin activity and attention. And immediately we wondered whether this could be causal. To test that, we can just perform a similar experiment, but now we're not using a protein that tells us about the serotonin activity. Instead, we're going to put a protein into the serotonergic neurons that allows us to manipulate their activity. Those two proteins are called channelrhodopsin and halorhodopsin. If you shine light onto channelrhodopsin neurons, they become more active. If you shine light onto halorhodopsin neurons, they become less active. You can inhibit them. And so what this allows us to test is exactly the control of serotonin on attention. The prediction then is that as you excite raphe neurons, attention should go down because you're getting rid of this raphe low state. And detection should, as a result, also go down. Whereas if you suppress these neurons, the opposite effect is expected to happen. You get an improved or higher attention towards cued features and locations and higher detection. The way we then execute this protocol is that we perform these manipulations just before the grading is scheduled to appear. Remember, mice don't know in this time period when it appears, but we simply activate or suppress neurons now. And when you excite these neurons, mouse performance goes down, as both characterized by a decreased discriminability index and a higher threshold. Whereas if you suppress these neurons, you get the opposite effect, higher discriminability, lower threshold. Now, one thing that you kind of get for free because of the way this task is structured is the following, which is that mice can still false alarm in this time window just before grading onset. And when that happens, we restart the trial. Oh, I have to say this, but okay, sorry. We restart the trial. So what you can get is false alarm links where the LED was on and false alarm links where the LED was off. Just by the simple stochastic nature of when might a false alarm link occur, might it happen during the stimulation window, or might it happen outside of it? And so this gives you the nice conditions of false alarms where you've manipulated raphe activity and false alarms where you haven't manipulated raphe activity in the same trials. So in a control mouse where you perform none of these, attention looks very normal. It's favored towards acute features and locations like verticalness and contrast. And if you then excite these neurons, that relationship falls away. Basically, no attention towards acute features and locations when compared to the uncute side. If you then release that inhibition, the relationship comes back very slowly. What didn't change again is whether or not mice used verticalness or contrast features. In all of these conditions, they used them. But what changed is the weighting towards acute features and locations. Mice used Q more when you don't excite the raphe neurons. And this is also indicated by a dropped attention index, which basically drops to 0 when you excite these neurons, and then it comes back up when you release that excitation. If instead you suppress the neurons, you get the opposite effect. Increased attention to acute features and locations when you suppress raphe neurons. And as you release that suppression, attention goes lower again, as also measured by this attention index. Now, it seems that we have this causal relationship of how serotonin seems to regulate how much attention are mice placing on acute features and locations. But everything I've shown you so far lives only within the entirety of the raphe. As I've kind of shown before, the raphe tends to project to many different parts of the brain, including the prefrontal areas and the visual areas that we care about so much. So because we were in a visual neuroscience lab, we decided to understand, do these types of signals that gate attention make it all the way to the visual cortex, where these types of gain changes of visual cortical neurons happen historically? Now, the way we can test that is we perform the experiment again, but use a serotonin sensor that we inject into the mouse visual cortex and then place a window on top of the brain and image through it while the mice perform this task. What you again get is signals where 5-HT is low just before grading onset or high just before grading onset, that you can again group into trials both containing hits and misses, where 5-HT or serotonin is now low, or 5-HT is high. You can compute the psychometric curves again, showing again that when 5-HT is low just before grading onset, performance is higher, especially at low coherent stimuli where attention helps out, stimuli that are hard to see. This is also characterized by an increased discriminability index when 5-HT is low, and a decreased threshold, indicating more sensitive to changes in grading coherence. What didn't change is the response bias, how much are mice guessing, or the motivation, the lapse rate, across these two conditions. Again, we can perform our attention indices from the false alarms that are coming from this data. When 5-HT was low before a false alarm link, attention was high towards acute features and locations, much higher than when 5-HT was high before a false alarm link. This is also indicated by a higher attention index when 5-HT is low. So it seems that these signals make it all the way to the visual cortex, where they could have the effect on the visual cortical neurons. And what I've shown you so far, again, this attention task, right? Mice place attention to acute features and locations. And in the simple model of attention, what our data suggests is that it kind of happens in two ways, the way attention reaches in. First is the selection of which features are important. We call this attentional focus. Remember, in our data, it didn't change whether mice use contrast or verticalness. It just changed how much they allocated their sensitivity to the acute features and locations. And in that sense, it seems that what serotonin gives is not the focus of attention, but rather the strength of it. Now, up till now, I've only given you this very simple model of attention. And only in this kind of very simple sense, where 5-HT just controls the strength of how much do we use these features. But over the last 2 decades, more complicated models that are specifically targeted at understanding how these types of gain changes occur in the visual cortex have been developed. Now, one of these models, which is very influential, is the normalization model of attention, in which stimulus drive, like input, like visual input, drives visual cortical neurons, or something like that. Drives visual cortical neurons, such as the one on the left here and on the right screen. This is then boosted by an attention signal, like the attention field up here, which is then applied on only one side of the screen to try to boost the intended location and improve the detection of those stimuli. Now, critically, in this normalization model, both the attention field and the stimulus drive both contribute to a normalizing suppressive drive, which is right here. Now, this then normalizes the attentional boosted response to give rise to this type of gain change, where one population response is higher than the other one. So this is the type of gain change that I've kind of alluded to in that monkey schematic. Now, as it so happens, other work in serotonin field has studied what exactly does serotonin actually do to visual cortical neurons, and it tends to suppress them, akin to this kind of suppressive drive. So one way to interpret the very behavioral results that I showed you today is that what serotonin might do in the visual cortex is scale suppressive drive. When 5-HT is low, just before a false alarm leak, for example, suppressive drive is also low, allowing the effects of this attention field that boosts one side of the screen to be more dominating. And you end up with this high difference between the cued and uncued locations in the visual cortical response. Whereas when attention is high, suppression dominates, normalization dominates, and what you end up with in the end is two types of population responses that actually look very equal, effectively weakening the gain effect that you get in the visual cortex. And so in this way, the behavioral effects that I showed you today are consistent with some of these more classical models of attention and suggest a concrete role in how serotonin shapes the strength of attention, but not its focus. Okay, now I want to kind of take a different direction for a couple of minutes, which is that while I was performing this kind of work within the lab, I got increasingly curious, how do mice actually place value on these stimuli that I've just happened to show them? Vertical bars, vertical energy, contrast. So in other words, how does a mouse associate that these things are rewarding and place this value on them? Well, it turns out that parallel work in the Bake and No labs seem to suggest that serotonin actually controls or encodes prospective value and signals it throughout the brain. Now, in this type of scenario, value is usually framed in this kind of task framework where you have this conditioned cue response. So here, the cue tells the mouse exactly when the reward is going to come. Then value increases as you approach the reward and you receive it. And then as you get the reward, value drops because now you've received your reward. So value in this case means how much reward am I getting in the future? This is very strong in contrast to the attention task that we have here, where the cue doesn't tell the mice when the reward comes in. It tells them where should they look. The actual time uncertainty and when the reward comes in is really, really high. And so this kind of contrast between what does serotonin do in attention and what does serotonin do in predictive value encoding is kind of raised some questions with it. First off, well, how can it encode both? What does serotonin, can serotonin kind of encode different contexts? What would serotonin do when a mouse is faced with two differently valued decisions and had to choose between one of them? And so to kind of work on these ideas, I've joined the Bake and No lab for my postdoc, and I'm now working on tasks like this virtual reality task, where a mouse has to navigate and make a choice between two arms. One is kind of this leaf arm that is associated with, I'm going to get reward really soon, and one is this grass arm that's associated with, that's not going to give me reward really fast. And you can kind of see how the mouse contemplates, then goes through this reward patch and gets its reward. What does serotonin do as a mouse and it sits there, makes its decision, associates one value to the right arm versus one value to the left arm. And then in a very different context, how would serotonin encode value in a very uncertain environment where only one of these 100 holes that seem to be hard to see, but only one of these 100 holes contain a reward, and now a mouse has to forage through this maze to try to find the reward. Here, a mouse is more likely to have to titer really nicely between kind of exploit or explore state. Exploit meaning, I'm going to go on the data that I've previously learned as it goes through many of these trials, and then explore, locally check where might the reward be. You can kind of map these types of states using hidden Markov models, as indicated by the colors here. So for example, now it's entering that blue state where it goes into this explore mode, tries to look at all of the holes that it have the reward until it finally finds one, and then flees back to this home cage. So those are the types of questions that I'm going to work on in the future. For now, I'd like to, first off, thank all the lab members of the Krishnaswamy lab and the Cadre lab that have made my PhD such an awesome experience. And of course, my 3 mentors that have provided me with so much mentorship that I'm now realizing as I'm entering my postdoc and setting up experiments for myself, but that I also get to pass on to the new students in the big lab. And then lastly, I'd like to thank you for your attention and take any questions you might have.
Thanks, Jonas, for a good talk. Questions?
Oh, wow.
Yeah, go ahead.
Great talking to us.
Thank you.
Glad to see you again. Never asked other members in the lab. I've never asked you. So, what's the reason for using water as the reward, as opposed to, if someone need to give them ice water, anyways, or they'll get hydrated and die. But why not go for a reward that's not necessarily something they need for survival? Let's say.
Why give them? Well, one thing is, do any type of behavior task, you need some motivation, right? So in this type of behavior task, the mice were likely water restricted, so they're pretty thirsty. So it's very easy to control how much are you motivated for that. It's really hard to otherwise say what motivates the mouse because you can't really peer into its mind. But yeah, you could use other rewards. So people would prefer, like when we actually started this, we were using Gatorade, so like a sugary reward. Now in the bake lab, we use chocolate milk. All of these type of things work. Water is just really experimentally to That's one of the things, yeah. But what else rewards mouse? Start having to interpret it all.
Actually, not well, the other one. Because I know the chocolate milk and stuff. Then you have to, there's nutritional value there to account for that. You thought of like non-caloric sweeteners and stuff. So it is sweet, but it doesn't change their intake most of the time either.
You mean like some aspartate kind of stuff?
Yeah, sure.
I haven't tried that. I wonder if somebody's done that. Oh, actually, well, that has been done. So there's some studies that try to directly measure how much is like a metabolically required reward, encoding reward, versus something that is more like what you're describing, like aspartate. So that kind of thing, I think it lives more in the hypothalamus, those types of systems that associate how much reward there is. But I think there's like actually a recent science paper that goes directly into that. And they say that, oh yeah, sugary reward, there's a group of neurons that's like sugary rewards are great. Aspartate is not that great. But then it's also, you get into that gut neurophysiology as well because of feedback signals. I don't know if that exists in the serotonin system so much. Yeah, there's a question. Yeah.
Go ahead.
Forgive me if this is a silly question because I've seen this library results before, but is there a reason why you only use vertical gradings? I feel like I should have asked this question like 4 years ago.
Well, the reason I use it because it started working. But the second one is no, I mean the lab is also working on trying to discriminate between different features. So I think, well, Eric actually told me about this on the way here. It's like you use vertical bars versus horizontal bars, and you cue them differently, and like can they discriminate those two different features? yeah, you can do those types of
Okay,
experiments.
so they can still discriminate if it's horizontal.
Say it again?
They can still discriminate if it's horizontal, and then they would.
Oh, yeah.
Like the attention, like tuning would be horizontal.
Yeah, that's the idea that I think
Okay.
the lab's testing now.
Okay.
Like, can you discriminate between different features? Right, because technically data doesn't say, oh, maybe they can't see horizontal, but
No,
they sure can.
Can I ask you about the vague supermodel at the end?
Sure,
The predictive coding one? Nice talk, but,
thank you. The
so-
supermodel.
yeah, so it's true that time is uncertain in the task, but after the cue, for sure, right after they false alarm lick, immediately afterwards, 3 seconds or something like that, definitely nothing happened. So should there be a ramp? Plateau?
Hmm,
What do you think should happen?
I don't think so. I think, well,
Just that blue thing kind of looks like the false alarm response.
there's a lot of these, one thing about why this is called perspective code is, so this is like a value function that you define through reinforcement learning, where at the start, right, value is now somewhere distant in the future, and then as you approach it, value becomes higher and higher. You're like, oh, I'm on the right track, I'm gonna get my reward soon, and then you get it. And what this perspective code does is it says through a heavy spiking adaptation. Oh god, I forget the spike frequency adaptation. I think needed to look at MR and so with spike frequency adaptation, right? You have an increase in value function that will give you this nice serotonergic response. So this is in line with many of the Q type responses that you would record in serotonin. And then you get this reward response, which might be surprising in other contexts, like, oh, I didn't know it was coming, then you would get like a larger response. Or if it's really expected, you would just drop the response. Now, one of the things that I'm kind of curious about is, and maybe don't fully believe, is that most of these types of value signals depend on an internal model, right, of the mouse. So it assumes that the mouse knows, oh, in 2-3 seconds, I'm going to get my reward. But how accurate can that model be? And I mean, you know, Paul Massey's work, trying to have different discount factors in the future. I think when you're getting into the paradigm of our task, where trials can often take minutes, first of all, that value assumption, that modeling of the world might not be as accurate. The second is that I'm not sure whether serotonin neurons actually encode value that cleanly, or how different the spike frequency adaptation is. For example, started recording in this task. I just don't have the recordings in the slide yet, but I'm recording 5-HT in the prefrontal cortex with that fiber there. And then you would see the response only ramping up kind of when the mouse starts approaching that grading stimulus. Now, timing of that doesn't matter so much. It seems to me more that it's like a distance thing. Does it know, can it make the model of the world where it knows, oh, I'm going to get my reward soon? How soon? Maybe it's 10 seconds, maybe it's 5 seconds, but it's, in this case, it's also locked to distance run. I think it's a bit more linked to that. How certain can the model be? Which is kind of why in this task, you don't see ramps. Even though the mice here are super trained into into knowing that approximate thing is the reward location, there's still an incredible amount of uncertainty in here just because there's like 100 holes. So these types of green states are like, oh, I'm going to go to my approximate location, and then the mouse maybe knows it's got to be somewhere in here, and then it has to start checking. So it got to go into this explore start. But which one of these holes is it going to be? Totally uncertain. The timing is not given. I think that's kind of the problem with how well can you prospectively encode value.
So will the prospective value part be the green part? I'm gonna make it to this in-between point, then I just have to check.
Yeah, that's a good question. I'm still trying to answer. I don't know yet, but you might think that, yeah. What you describe as explore and exploit is a little hard to define there, but yeah.
Yeah, you got memory in it. So you have in the beginning you showed it, you had like your false positives, right? With noise with high vertical energy from the false positive. Is there like memory? So the I guess the probability of false positive is higher or lower depending if it was one closer and nearby or not, or a real signal. I don't know if my question makes sense.
So the idea is you get a stimulus.
Yeah.
And you have to hold it in memory and then kind of assign value.
Yeah, well, if I guess the probability to get a false positive increases or decreases depending if there was a stimulus close by or not.
Oh, I mean, yeah, it's hard to say whether that's in the attention task, it's hard to say whether that's memory or whether that's more like stimulus decoding, because there's always like a reaction time involved and whatnot. And most of these false alarm links, I think I have the plot somewhere here. Most of these false alarm links, they do happen pretty close by, right? Like 300 milliseconds. So that's in line of the most of the reaction times that mice will do in this. So I doubt there's all that much like memory in that sense here. But memory is an interesting question because I mean, some memory is involved, right? Like long-term learning is involved. You have to assign credit to these verticalness, and clearly mice are doing this, otherwise these feature maps would never develop. And I don't have clear data on this, but I'm kind of working on a different variation of that virtual reality task, where instead of deciding between two contextual differences, we kind of make it explicit, like, oh, I can't tell you.
You have to take your red
Yeah.
dot off.
Right? Instead, you would give them the cue here, go leaf or grass. You'd have to keep that in memory as you approach that intersection. Then you'd have to make a choice. Am I going to go leaf or grass? So you'd have to hold it in memory, and then you can maybe see it, how serotonin would have to encode value in that sense. That's kind of an idea we're working on right now. But it happens to be pretty difficult because when you train something, when you train an animal, you kind of have to tell them that thing you just did, that was rewarding. And then when the time difference becomes so large, the credit assignment becomes pretty hard. But yeah, it's a good question. I wish you can do that.
There's a question in the chat if you want to read it.
Oh.
You want me to read it?
Sure, yeah.
Okay, it says, you could try putting the reward spout to the left or to the right and have the reward appear predictably, left and right. Suresh, you want to explain your idea? Sorry.
No, I'm just thinking through and following up on the idea. So serotonin is low when you're paying selective attention to one side. During the reward, you're paying attention to the center, not biased. And you're kind of shutting off, putting up your serotonin and avoiding sort of attending either way. So if you make the animal attend during that period, I'm just curious as a simple control. If the reward is coming from the left and the right, during the reward period, the animal will selectively attend to the left and the right because the reward is going to come from one side. So I'm just curious if the selective attention during the reward waiting phase actually produces a predictable effect on the serotonin.
I mean, they do maintain that strategy, right, during very early licks during the actual reward presentation. So I think I showed this here. Right. So in the center one, you're taking false alarms during the actual grading and looking at the checkerboard noise that preceded the actual reward, the actual grading onset. And so during those licks, even when the grading is on, they still take the same type of strategy where it's one-sided, and they maintain that throughout the entirety of the task. So they would still attend towards the cued features here.
No, but in your new task, in the prospective coding theory, the prospective coding theory, you're waiting for the reward to come from the front.
Yeah.
I'm saying that if you instead attended to one side or the other, while waiting for the reward, presumably serotonin may do something different because it's now a biased attention.
Yeah. I mean, yeah. That's exactly what I'm trying to understand. Which I'm not sure how this different spouts would help you. I mean, you can because they don't have to actually make.
They have to lick, right? You have to lick towards the right or to the left if it's coming from the side. So it's not.
What do you mean at the decision point?
Yes, exactly. So they are waiting for the juice to come from the left or the right. And it's a mild tongue turn. I'm sure they can twist their tongue slightly to receive reward that's coming from slightly off to the side.
Yeah, I've thought about doing that. So you basically have two spouts in front of it. and then they report their decision on what to do based on what spot they do, and then it
Yes.
locks them kind of in one of the
Yes,
two.
exactly. And this is now spatially biased lateralized. And then the idea is that the serotonin is kind of controlling the level of spatial bias in the attentional signal.
Yeah, you could do that. Couple of downsides is that you kind of get rid of some of the navigation stuff that we're also trying to look into.
Yeah.
Yeah, you could make it two alternative voice choice in that way. Yeah, that's cool. Yeah.
Yeah, it doesn't even have to be a real task. It could simply be
Oh, sure.
predictable. Simply predictable in that they will automatically choose. They know where the reward is coming. So effectively they will attend to the left to pick up the reward. So it's a predictable thing. So if they hit in response to the right, or you can set it up in many ways. But anyway, this
Yeah. Okay. Yeah, I'll think about it a bit more, but I think it's a good idea.
yeah.
Thank you.
Stock.
I miss this for the next slide, but for the maze task, how did the mouse know to start checking near the area of the reward, or is that just random chance?
It's kind of a learned strategy. So in the beginning, they know nothing. They're just like in this, all of a sudden in this huge corn maze, and they have to search for the one hole that has the reward. So they start just like exploring the entire time. Then you do this over a couple of trials, a couple of days, and they very quickly learn, oh, I just got to dart to this one direction roughly, and it's completely egocentric, like it's completely from within that they now have to estimate where that rough location is. And then it seems that the further it's away, the more they're gonna start checking, because I think it's because their uncertainty grows as they leave their home cage, and then they're like at some point the uncertainty dominates, and they're like, okay, I just got to start checking. This is the most efficient way to do this. It's like really like policy kind of refinement is like, oh, I'm going to make my exploitative decision, and then the low cost next policy would be I'm just going to start hole checking. So that's kind of how they do this, and this is kind of what I'm trying to capture with Markov models, is just through their type of different behavior. Right, these colors are from a Markov model itself that's trying to look at their posture, look at how much are they turning, and some contextual data. What kind of behavioral regime they're in, to see like, am I in like more in an explore state, and then you can compare serotonin dynamics there. Am I more in an exploit state? So you can compare there.
So what is your goal by doing this hidden Markov model, like detecting more hidden Markov models?
Well, it's pretty hard to hand label, right? In this case, when am I in exploit? So I'm trying to make a unified algorithm for all the mice, where that tells us in what behavioral regime are you in. So the hidden states are things like, am I in an explore state or am I in an exploit state? Of course, because it's hidden ones, it's totally up to interpretation. And then also throwaway things, like maybe things that I don't care about yet, like this red state is that's down here. It's kind of like a grooming state where the mouse is not moving much and it's just like grooming. So I'm trying to label them and then show what happens at the state transitions in the 5-HT signal. Because at some state transitions, it would be, okay, I've transitioned into this very uncertain environment. How does serotonin signal that?
I see. Any other question? Chat? Okay, all right. Thank you very much, Jonas, for a great talk.
Thank
And please.